Parkinson's and other neurodegenerative diseases are among the most urgent health problems worldwide. These diseases cause neurons in certain areas of the brain to degenerate. They are still incurable, and as has long been known now, they do not only affect the elderly. So how can we speed up the development of new therapies? One promising approach, which experts in Luxembourg are now taking, is to do research on the patient's own cells. It starts with the preparation of skin samples at the Integrated Biobank of Luxembourg. Small tissue samples donated by Parkinson's patients are placed immediately after being taken into a nutrient solution. Over several weeks, the individual skin cells in the samples multiply. They are then frozen in liquid nitrogen at -196 degrees, putting them into a state in which they can be kept viable and available for research at any time in the future. The scientific work begins at the Luxembourg Center for Systems Biomedicine of the University of Luxembourg. By adding certain factors to the skin cells, the researchers set them back into a so-called pluripotent state, where they can give rise to all tissues of the human body, but never a full human. Just like embryonic stem cells, but without the ethical implications. The advantage of such stem cells is that they are undifferentiated. By addition of a growth factor cocktail, they can once again differentiate into nerve cells, neurons of the specific type that are affected in the brain of Parkinson's patients. Cultures of these nerve cells offer ideal conditions for testing drugs that could help fight the disease. The LCSB has installed a worldwide unique, fully automated system for this very purpose. Latest generation robotics allow a whole library of drugs to be analyzed simultaneously with much greater precision than by conventional laboratory techniques. This is important progress because despite intensive research, only few successes have been recorded over the past decades in drug development for Parkinson's disease.
Currently, medications for Parkinson's disease patients are developed on animal models or on cellular models that are far away from the actual tissue that is affected in Parkinson's disease. With our strategy, we use neurons from the patients to test the compounds on the tissue that is actually undergoing neurodegeneration in Parkinson's disease. And this is a unique concept. This is only feasible having an automated system that guarantees a high constant quality of the different procedures to find out this single compound that is then available for reverting the symptoms and that helps us to bring back something neuroprotective to the patient.
The researchers, however, also want to better understand the complex processes in the brain that lead to the onset of the disease. In a special culture, they allow patient stem cells to grow into three-dimensional neural structures that resemble little brains, which are therefore referred to as mini-brains. Under a fluorescence microscope, the areas within it that are affected by the diseases can be targetedly visualized. This way, the researchers can investigate how the changes typical of the disease arise in the brain.
We compare these mini-brains from a Parkinson patient to those from a healthy individual and try to find out all kinds of things that are different between those. And as soon as that is identified, you can think about screening for drugs, for compounds that have the ability to normalize the disease situation and therefore could lead to new therapies.
To optimize the work in the lab, the LCSB also employs methods of computational biology. Using sophisticated algorithms and mathematical models, they can predict, for example, how the transformation of one cell type to another cell type can be made more efficiently. Computer models can also reveal new strategies for correcting the disrupted gene expression program of affected cells without having to correct disease-related mutations, a way to slow the progression of the disease.
If we look at disruptions of gene regulatory networks due to specific mutations or disease-related mutations, it would be kind of similar to the fact if you look at a city and you have some streets or highways blocked. The question there is how to restore the circulation, the traffic, without actually unblocking those streets.
Parkinson's is a disease that develops gradually. The researchers are therefore cultivating neurons over relatively long periods in another specially developed system. In it, 96 cell cultures in tiny channels can be tested simultaneously and fully automatically. The aim is to better understand the chronology of the disease's many different symptoms.
This is a 3D print, 20 times magnified, of the inside of a microfluidic cell culture device that we use here. The cells come in here and they stay on this side, and the food for the cells passes on the other side. It's designed for studying over a prolonged period of time the same cells and repeatedly observing them. In Parkinson's patients, when they present in the clinic, they often have multiple changes, but you'd like to know which came first and which came second. And if you find which came first in the series of events, you have a chance of trying to slow down or stop the progression of disease.
Understanding Parkinson's disease better and at the same time testing treatments on patient cells to develop new therapies. The combination of these approaches makes the LCSB a leading center for the research of neurodegenerative diseases. An outstanding example of interdisciplinary cooperation in healthcare in the research location Luxembourg.